Mutations and genetic technologies might seem like separate topics, but in reality, they're part of one big story about how we understand and manipulate DNA. In this revision session, we're going to break down Module 6 in a way that helps you see how all the key ideas connect, from how mutations create new alleles to how scientists are now using CRISPR to edit DNA itself. Let's jump straight in. If you've ever wondered why no two people look exactly the same, or why some species thrive while others go extinct, the answer lies in mutations. At the most basic level, mutations are the source of all genetic variation. Without them, evolution wouldn't happen. A mutation is simply a change in the DNA sequence, and while the word often has a negative connotation to it, not all mutations are bad. Some mutations can cause genetic disorders or diseases like cancer, but others include beneficial traits that give organisms an advantage. Over generations, these beneficial mutations can spread through a population, shaping how species evolve. There are a few key ways that mutations arise. Some happen naturally due to errors in DNA replication, where the cell accidentally inserts, deletes, or swaps the wrong nucleotide when copying its DNA. Other mutations occur because of external factors like radiation, chemicals, or even viruses that can alter DNA sequences. These changes introduce new alleles into the gene pool, creating variation that natural selection can act upon. For example, let's look at sickle cell anemia. This disease is caused by a single point mutation in the gene that codes for hemoglobin, the protein in red blood cells that carries oxygen. In individuals who inherit 2 copies of the mutated gene, the result is sickle cell disease, which can be life-threatening. But in individuals who only inherit 1 copy, this mutation provides resistance to malaria, a deadly disease transmitted by mosquitoes. In regions where malaria is common, natural selection has favored this mutation because it helps people survive and then reproduce. This is a perfect example of how mutations aren't always bad. Their impact depends on the environment. What might be harmful in one context could be beneficial in another. Another example is antibiotic resistance in bacteria. Bacteria reproduce incredibly quickly, and random mutations in their DNA sometimes result in a small number of bacteria becoming resistant to antibiotics. If a person takes antibiotics but doesn't finish their full course, the resistant bacteria survive and reproduce, passing on the mutation. Over time, the population of bacteria becomes entirely resistant, making the antibiotic ineffective. This is why new antibiotics constantly need to be developed. It's an ongoing battle between human medicine and bacterial evolution. The key takeaway here is that mutations drive genetic diversity, which is essential for evolution. Without mutations, every organism would have the exact same DNA as its parents, and species wouldn't be able to adapt to changing environments. The next step in understanding this process is looking at what happens when a mutation affects DNA, how it changes gene expression and then leads to new traits and diseases. Let's break this down. Gene expression is the process by which DNA is used to create proteins that determine an organism's traits. Mutations can occur at different levels, either affecting a single nucleotide or entire sections of the chromosome, and their impact depends on where they occur and whether they alter protein function. At the simplest level, mutations can be categorized into point mutations and chromosomal mutations. Point mutations occur at the nucleotide level. They involve a really small change in a base pair of DNA. These are the 3 main types. The first one is silent mutations. The mutation changes at a base, but the resulting protein stays the same. This happens because multiple codons can code for the same amino acid, meaning the mutation has no real effect. Another one is missense mutations, which is where the mutation changes a base, which then leads to a different amino acid being incorporated into the protein. This can have a mild or severe impact depending on whether the change disrupts the protein's function. And the third one is nonsense mutations. The mutation can cause a stop codon to appear too early, cutting the protein short. These mutations usually have really serious effects because the protein never fully forms. A real-world example of a missense mutation is sickle cell anemia, where a single base change in the hemoglobin gene leads to abnormally shaped red blood cells. Meanwhile, cystic fibrosis can be caused by a nonsense mutation, where a protein important for mucus regulation in the lungs is cut short, leading to severe breathing difficulties. Chromosomal mutations, on the other hand, occur on a larger scale that involve changes to the entire sections of chromosomes. These include deletions, where a section of a chromosome is lost, which can remove multiple genes; duplications, where a section of a chromosome is copied, leading to extra genetic material; inversions, where a segment of DNA is flipped, which can disrupt normal gene function; and translocations, where a piece of one chromosome attaches to another, potentially disrupting multiple genes. A well-known example of a chromosomal mutation is Down syndrome, also known as trisomy 21, which is caused by an extra copy of chromosome 21. This results in developmental delays and distinctive physical features. Another example is chronic myeloid leukemia, or CML, which is linked to translocation between chromosomes 9 and 22, leading to uncontrolled cell growth. The location of mutation also matters. If a mutation occurs in a coding region, which is a gene that makes a protein, it directly affects protein production. However, if a mutation occurs in a non-coding region, such as regulatory DNA, it can affect where and how much a gene is expressed rather than changing the protein itself. This explains why some mutations have no effect at all, while others can lead to life-threatening conditions. It also highlights why scientists are increasingly interested in editing or repairing mutations using genetic technologies like CRISPR. Now that we understand how mutations affect DNA and gene expression, what if we could control mutations? Well, that's exactly what genetic technologies allow us to do. From treating genetic diseases to producing pest-resistant crops, biotechnology has revolutionized the way humans interact with DNA. One of the earliest and most widely used genetic technologies is recombinant DNA technology, which allows scientists to insert genes from one organism into another. This is how we produce genetically modified organisms, or GMOs, such as crops that grow faster, resist pests, or tolerate extreme weather conditions. It's also how bacteria are modified to produce human insulin, a life-saving treatment for diabetics. Another key breakthrough is cloning, the process of creating genetically identical copies of an organism. While cloning has been around since the 1990s, famously demonstrated by Dolly the sheep, it's still highly controversial when applied to humans. Cloning raises ethical concerns about identity, genetic diversity, and the potential for misuse of reproductive technologies. But it has major applications in conservation, where endangered species are being cloned to help restore declining populations. In my opinion, the most exciting advancement in genetic technology is CRISPR, a gene editing tool that allows scientists to precisely cut and modify DNA sequences. Unlike older gene editing methods, CRISPR is faster, cheaper, and more accurate, making it a potential game changer for treating genetic disorders. Scientists are already experimenting using CRISPR to fix mutations responsible for diseases like sickle cell anemia, cystic fibrosis, and even some cancers. But the ability to edit DNA doesn't just apply to medicine, it's also transforming agriculture and industry. Gene therapy aims to replace faulty genes with functional ones, potentially curing inherited diseases before symptoms even appear. Meanwhile, genetic engineering in livestock is helping produce disease-resistant animals, reducing the need for antibiotics and increasing food production efficiency. Of course, the ability to manipulate DNA has given us incredible medical breakthroughs and agricultural advancements, but it also raises major ethical, social, and economic questions. The biggest concern is where to draw the line. Should we only use gene editing to cure diseases, or should we also allow modifications for non-medical traits like intelligence, athletic ability, or appearance? This debate is especially relevant when it comes to germline editing, where genetic modifications are made to embryos before birth. Changes to germline DNA don't just affect the individual, they get passed down to future generations, meaning any mistakes could have permanent consequences for the human gene pool. Scientists have already used CRISPR to edit embryos, but most countries have really strict regulations or outright bans on germline modifications due to these concerns. Another major issue is access and inequality. Gene therapies and genetic modifications are expensive, and if only wealthy individuals can afford them, it could create a world where genetic advantages like resistance to disease or even cognitive enhancements are only available to certain groups. This raises the risk of genetic discrimination, where people with unmodified genes are seen as inferior or disadvantaged. There's also the question of who controls genetic information. With the rise of DNA sequencing, people can now get their genomes analyzed through companies like 23andMe. But this means private corporations are collecting massive amounts of genetic data. If this data is misused, whether by insurance companies, governments, or even criminals, it could lead to serious privacy violations. Genetic modification in agriculture and industry also raises ethical concerns. While GMOs have increased food production and reduced pesticide use, they also lead to controversy over corporate control of farming. Large biotech companies patent genetically modified seeds, meaning farmers can't save their own seeds and must buy new ones each season, creating economic dependence. The environmental risks of genetic modification are also huge. Once genetically engineered crops or animals are introduced to the wild, they can alter natural ecosystems in unpredictable ways. For example, genetically modified salmon, which grow faster than wild salmon, could outcompete native populations, disrupting entire food chains. The overuse of genetically modified pest-resistant crops has also led to the rise of superweeds and insect populations that have evolved resistance, forcing farmers to use even stronger chemicals. But despite these concerns, genetic technologies aren't inherently good or bad. It all depends on how they're used. Which leads me to my next point. How do these advancements affect biodiversity? And what are the long-term consequences for life on Earth? Biodiversity is essential for ecosystem stability. The more genetic diversity within a population, the better it can adapt to changes such as disease outbreaks, climate shifts, and habitat destruction. But as genetic technologies become more and more frequently used, they're starting to both help and harm biodiversity in unexpected ways. One of the most interesting areas of genetic engineering is conservation genetics. Scientists are now using cloning, selective breeding, and gene editing to help save endangered species. For example, efforts are underway to bring back the northern white rhino, a species that only has 2 living females left. Using reproductive technologies and stored genetic material, conservationists hope to prevent its extinction. Gene editing is also being explored as a way to make species more resistant to diseases, such as modifying frogs to withstand fungal infections that have devastated amphibian populations. But genetics is also being used to control or eliminate certain populations. One of the most controversial examples is gene drives, which allow scientists to push genetic modifications through an entire species at an accelerated rate. This has been proposed as a way to wipe out disease-carrying mosquitoes by introducing a genetic trait that makes them all sterile. While this could potentially help eliminate malaria and other deadly diseases, it also comes with massive ecological risks. If mosquitoes disappear or dramatically decline, it could disrupt food chains and harm species that rely on them, including birds, fish, and other insects. Agriculture is another area where genetic engineering is changing biodiversity. Genetically modified crops have been designed to resist pests, tolerate harsh climates, and produce higher yields. While this has led to increased food production, it has also reduced genetic diversity in certain crop species. Traditional farming methods involved planting a wide variety of crops, which meant that if a disease or a pest wiped out one strain, the others could survive. But now, large-scale monocultures, where only one genetically modified variety is planted, have become common, making crops more vulnerable to diseases or environmental shifts. Livestock genetic engineering has the same kind of risks. One of the biggest concerns is that once genetically modified organisms, or GMOs, are released into an ecosystem, there's no way to take them back. If a genetically engineered species outcompetes a native species, it could lead to unintended extinctions and biodiversity loss. This is why scientists are urging caution, carefully studying the long-term effects before making large-scale changes to natural populations. Ultimately, genetic engineering presents a paradox when it comes to biodiversity. It has the potential to save endangered species and protect ecosystems, but also has the potential to cause irreversible harm if mismanaged. The key is responsible use, regulation, and ongoing research to ensure the benefits outweigh the risks. That wraps up the discussion of Module 6. If there's one thing you should take away from revising Module 6, it's that mutations, genetic technologies, and biodiversity are all part of the same bigger story. Mutations introduce variations, genetic technologies allow us to control that variation, and the choices we make about using these technologies impact entire ecosystems. A lot of students fall into the trap of memorizing individual facts without considering how they interact with each other. But exam questions want you to apply your knowledge across these different topics. Instead of asking you to simply define CRISPR or describe a mutation, the HSC might ask you to explain how gene editing could impact biodiversity, or to evaluate the risks and benefits of reproductive technologies. That's why it's so important to see the links between these ideas. So now that you've revised the content, take it one step further. Test how well you can connect these dots. Can you explain how a mutation could lead to a genetic disorder, and how that disorder might be treated using biotechnology? Can you describe how a genetically modified crop could affect both agriculture and biodiversity? Being able to make these connections is what will set you apart in the exam. A great way to make sure your understanding is solid is by actively engaging with the material. Instead of just reading your notes, try to break down these concepts into structured revision tools. Start with mutations at the center, and then branch out to how they affect gene expression, how humans manipulate genes through biotechnology, and then branch out even further to how those changes influence biodiversity and society. The more you interact with the content, whether through flashcards, practice questions, or structured study guides, the easier it will be to recall in your exam. If you're not too sure where to start, Excel HSC Copilot has a range of flashcards, study cards, past HSC questions, video explanations, and instant response feedback to help you test your knowledge. These resources will help you refine your understanding, practice exam-style questions, and get comfortable with applying your knowledge under timed conditions. The key to mastering Module 6 is active revision. So use these tools to check your understanding, identify weak spots, and then build your confidence before the exam. So thanks for watching and good luck with your revision.